Today's lecture is going to cover Chapter 9 from the textbook, and I have just a bit of a diversion before we start that presentation. Most likely this presentation of the PowerPoint file for Chapter 9 is going to take two separate lectures. So we'll do the first today on this recording and follow up. What I want to do before beginning that presentation is to show you a link on the website for a relevant research article and a useful site. So we'll switch to the computer, put myself in the pip here, oops, and we'll go to our course materials under the course content here, and steroid receptors, the link number 6. I should move that up a couple, probably will for future use, since we discussed those first. And the first link, I'll take you to that in a second. That's the article, review article that is relevant for our presentation, either within this portion of the lecture or in the next, -2, Chapter 9-2. And then this is an interesting site here that has information on it, and you will see that I have utilized some of this information in the presentation. And of course, we begin our discussion with the steroid receptors by thinking of them as ligand-dependent transcription factors. We have stated that any protein that binds to DNA and affects transcription is a transcription factor. So with that definition, of transcription factor, we can further begin to appreciate those that depend upon a molecule to bind to them, a biologically active molecule to bind to them in order to activate them, and that's the ligand dependent. So typically it's an extracellular chemical messenger, such as the hormones and thyroid, the steroid hormones and thyroid hormone. The first of which we'll discuss today, the thyroid hormone mechanisms of action. We'll consider when we discuss the HPT in detail. We've already mentioned some other transcription factors that merely need to be phosphorylated and then interact with other activated transcription factors, forming either homodimer or heterodimer to bind to their consensus sequence response element or regulatory element on the promoter region of target genes. The steroid hormone receptors and their regulation by phosphorylation is the journal article here on this link. Still as of 2005, this 1996 article is coming up, and it has, let me enlarge this full scale, it has all of the figures that are relevant for our discussion, and very good and interesting reading. I encourage you to look it over. It is a bit dated, but no problem. In this course, we are appreciating what's in the textbook, what appears in review articles, and what appears in primary literature. And we'll have a few recent articles, more recent than this 1996, and then a few older primary articles, just their abstracts, presented in our discussion through the Chapter 9. So let's go to our PowerPoint presentation. We are considering Chapter 9, steroid and thyroid hormone receptors. Well, there should be another E in there. Thyroid receptor and mechanisms of action of thyroid hormones, as I said, will be discussed in a subsequent lecture when we, setting my arrow here, sorry, when we look at the HPT axis in detail. So we're focusing just on the steroid receptors. However, we are thinking about the intracellular receptor superfamily that includes the thyroid hormone receptors, as you see on the list down here, as well as others such as vitamin D. So we begin all-inclusive and then narrow in on the steroid receptors. So there are two classes of this nuclear receptor superfamily, and of course this is taken from Table 9.1 in the textbook. The Class 1 nuclear receptor superfamily, and they are intracellular, and they target the nucleus. They may not reside in the nucleus. Let's just state it this way. They can reside either in the cytoplasm or in the nucleus, and we'll get into the specifics of where each particular type of receptor resides in the unligated form as we proceed. So they are intracellular because these are steroid, lipid-soluble chemical messengers, or in the case of thyroid hormone, it is transported across the membrane, as we will discuss. Regardless, they enter the cell rather than, or in contrast to the water-soluble peptide hormones that we've been discussing, and even neurotransmitters, classical neurotransmitters, that must bind to the extracellular portion or domain of its cognate receptor and have transmembrane signal transduction. These chemical messengers get right to the nucleus, and primary mechanism of action is effects on the gene expression, rate of transcription predominantly in the target cells. As we'll see at the end of this discussion for this chapter, as far as neurons are concerned, there can be some immediate effects, and we'll try to understand how those might be happening, because there's been no other type of functional receptor yet identified, and if it is there in the literature, the number of papers are not numerous. I did a little bit of searching today and nothing popped up. So there's still question about those receptors responsible for the more immediate effects of steroids on neurons. But from now on, until we discuss that specifically, we're thinking about targeting gene expression in the target cell, therefore nuclear receptor superfamily. So the class 1 are the androgen receptors, progesterone receptors, also called progestin receptors, glucocorticoid receptors, and the mineralocorticoid receptors. And then the class 2 are the estrogen receptors, thyroid hormone receptors, retinoic acid receptors, and the vitamin D receptors. And this classification is based upon the similarities in the amino acid sequences. So all of these hormones are proteins, hormone receptors, are proteins. They have been identified, isolated, purified, characterized, sequenced, and so on and so forth. And the genes have been identified, characterized, sequenced, and investigated, as well as the promoter regions for most, if not all of these. I'm not saying we know everything about them, but we know a lot about all of them, all the way to the level of the gene, and whether or not there's alternative splicing for the control of transcription. We know more and more about the post-translational processing of these, as well as the gene regulation at the promoters. So there's a wealth of knowledge about all of these receptors. Long discussion, just reminding ourselves that the proteins have been sequenced, and it's based upon the similarities in the amino acid sequences by which these have been classified. You'll notice that in this chapter and some of the other chapters, different methods have been described or mentioned, and I have this slide here to remind myself to inform everyone and remind you that we have a dedicated section of the course to discuss in greater detail many different methods utilized in the study of neuroendocrinology and endocrinology, and of course many other areas of investigation as well. So we just have a brief comment here and an illustration of how it would look, and then we'll talk about these methods such as autoradiography for the tritiated ligand, the tritiated synthetic hormones, how and why we can answer as we proceed. And immunocytochemistry has emerged since the classical autoradiography, using radio-labeled molecules. And in this case, the immunocytochemistry obviously uses antibodies to bind to and identify and localize and visualize the receptor proteins. So a difference between these two would be that the autoradiography for the radio-labeled ligand would identify the protein receptors that are capable of binding the ligand, the cognate ligand. Immunocytochemistry is only identifying the receptor proteins that can bind an antibody. So neither of these are a functional assay, and there's limitations. You may have an antibody that would stick to a dying protein maybe, and you'd have some image or quantitation of that protein in your pool that would have nothing to do with whether or not it could bind the ligand or even produce a response in the target cell. But we have serious questions, as you'll see, about which cells contain which receptors. And the anterior pituitary will be one of our focuses in that we ask the question, do anterior pituitary cells contain the androgen receptor that we think of for testosterone, or do they contain the estrogen receptor in both in both genders, and how about in humans? So this is a key question. If the anterior pituitary cells, say for the gonadotropins, the gonadotrope cells contain only the testosterone androgen receptor, then how do we get negative feedback in the female where the predominant gonadal steroid hormone is estrogen? If it's only the estrogen receptor, then how do we get the negative feedback in the male? So these are serious questions, and we'll answer that to some degree in this discussion of Chapter 9. Might show up in the Chapter 9-2 lecture. In situ hybridization is another method we'll talk about. All of these are related to having one molecule that you have that can bind to a molecule in the tissue and of interest in the tissue, and you can somehow visualize it. Hybridization makes us think that it's a DNA or RNA, and indeed it is a hybridization of either cDNA to the messenger RNA in the cells, or cRNA to the RNA and messenger RNA in the cells. Then we can determine what cells contain the message for the receptor, and thereby we feel confident that those cells express that gene and that that message is translated into the protein, and of course that it's a functional receptor. So if you identify it, it doesn't answer all the questions. Does it? In the case here where we identify the cell that contains the messenger RNA, does it translate that? Is it regulated gene expression? Is the translated protein functional in that cell? And so on and so forth. So we always have to think about the limitations of the information we get from any one particular method. And that's a skill that we want to develop a bit in this class. Here I give you the source for this figure, and it is a combined dry mount autoradiography for, now in this case the animal was injected with the radio-labeled hormone, in this case tritiated estradiol, and then euthanized, and the tissue was taken excuse me, and then sectioned and visualized through these methods. The autoradiography, you have to put some type of photographic emulsion. In this case, they just put the emulsion right on the microscope slide over the tissue, and then exposed. You can see here, I think this one was exposed for 8 months, so very slow to collect the data. But you expose it for 8 months, just like you expose a negative in a camera that has, not a digital camera, but the old kind that had the film with the emulsion on it, and the light causes the precipitation of silver particles. Then you have to develop that, and then you can see the silver particles either with bright field illumination, where you're just on an upright microscope, you let the light come through, and you focus it, and you can see the little silver grains scattered all around, and especially concentrated on the nucleus of this cell. And it is counterstained so that you can see all of it. In this panel, and these are neurons in the brainstem, I think from the A1 location in the brainstem, staining for catecholamines using an old method called formaldehyde-induced fluorescence. The catecholamines, they have that catechol ring, so they have some capabilities. If in the presence of formaldehyde and appropriate illumination, they will fluoresce, or activation. So you see the co-exposure here, where you have the dark field illumination. You just kind of bend the light so that if it goes to the tissue, it misses and doesn't come up into your microscope. But if it hits a silver particle, then it bends and goes up into your microscope. So now the black looks white and the white looks black. So it produces a negative image. As well, you have the glowing cytoplasm of this neuron for the catecholamine itself. Remember, we talked about catecholamine synthesis in prior lecture and said that these neurotransmitters can be synthesized in the soma, the cytoplasm of the soma, as soon as the enzymes and the precursors are available. So here with the fluorescence, we see the catecholamines right in the soma of this neuron. It's not such a sensitive technique that we see all the dendrites and axons, but that's fine. We can identify the cell body, the cytoplasm here, and then you can see the nucleus doesn't contain the catecholamine fluorescence, but it does contain all these white dots, and they are concentrated there. Why are they concentrated there more so than just floating around out here in the neuropil? Because this is where the receptor is for estrogen. The estrogen receptor is in the nucleus, so it has ligated the tritiated estradiol, holding it in place. Of course, the formaldehyde helps to fix everything, cross-link all the proteins in place too. But it is concentrated there. So you've injected the animal, give some time for distribution throughout the body, and binding to receptors, and thereby the accumulation in the nucleus. And then when you look at just the fluorescence for the catecholamines, here now you can begin to make out some axons and maybe some dendritic processes, but not as sensitive as the immunocytochemistry. But what you note is that when you look just at the fluorescence of the catecholamine, of course there's nothing in the nucleus. That's how we know this is the nucleus covered with the silver grains for the tritiated estradiol. So they are nuclear concentrating receptors. They concentrate the estradiol, in this case, in the nucleus. So these are noradrenergic neurons in the brainstem. Other methods have validated that these catecholamine neurons in the brainstem are indeed noradrenergic, and they actually do express the estrogen receptor gene. The estrogen receptor protein is present, and I'm not certain if any functional studies have been done to demonstrate the estrogen receptor is indeed functional in these. But we'll talk about these noradrenergic neurons in the negative feedback pathway when we talk about the HPG axis. This slide is showing the concentration of vitamin D, tritiated 125 dihydroxy vitamin D3, and the immunostaining is for the beta subunit of thyroid stimulating hormone. They're showing the dilution of it here. So there's other methodological detail in these legends, and I encourage you to pay attention to them now, but we'll develop our skills to recognize and understand the significance of those. The one thing that's missing from this figure in the prior one is some type of a bar scale so that you know whether this is 10 micrometers, 20 micrometers, or whatever. So usually there's a little horizontal bar with a scale for it. This is the anterior pituitary. Obviously, these are thyrotropes. You see the staining in the cytoplasm. In this case, it's a color product or a chromogen that's deposited with the immunostaining method. So these are thyrotropes. And you see that none of the TSH beta, in this case, beta subunit, is within the nucleus. We wouldn't expect it to be there. If it was there, we'd question what we were staining. So the nucleus is unstained, but we can see, again, the condensation of the silver grains representing the tritiated 125 dihydroxy vitamin D3. This one was exposed for 6 months, so somebody was very patient to get the results for this. But it demonstrates very nicely how these receptors can be visualized. This looks like vitamin D in a capillary here, so they probably just removed the pituitary, didn't get all the blood out, and that just looks like the vitamin D entrapped within the capillary. We just see it, and then it's scattered throughout the interstitial space. And then other than the plasma, the only place that it's concentrated is within the nuclei of cells, and it looks like it is predominantly in the thyrotropes in this particular image. Well, we can see it there. So we've talked about expressing the gene, having regulated expression of the gene for these receptors, the transcription, RNA processing, is there any type of alternative splicing, the translation, post-translational processing, and mechanism of action. How does it act? How is it activated? And obviously there has to be a degradation, and these are proteins, so when they are old and worn out, they're probably removed from the cell by the ubiquitin system. Ubiquitin system labels proteins with ubiquitin, and then they're targeted for lysosomal or intracellular enzyme destruction, recycling of amino acids. So we're keeping in mind this life cycle of every molecule that we talk about, from regulated gene expression to control synthesis, the life cycle from the inactive pool to the active pool to biological action, biological inactivation, and either recycling for reactivation or degradation to get rid of it. So it's the standard model for the life cycle of molecules. Then we ask the mechanism of action as we proceed. So these are hormones with intracellular receptors. They're either the thyroid or the TH hormones, the steroid or thyroid hormones. They're in the cells, either in the cytoplasm or the nucleus, and they function as the ligand-dependent transcription factors. So you see this is from that site that I give the source up here, and the link I've showed you at the beginning of the class. It's a very nice site. I like the way they present the information, obviously, and I've extracted it for our presentation. So the hormone receptor complex. So remember, this is another term for this that I will use is the ligated receptor. So the ligand is the chemical messenger or the synthetic or exogenous molecule, and it sticks to, or like we ligate an incision in the skin, we stick it back together, and the ligand ligates the receptor, and the ligated receptor becomes complex, either homodimeric or heterodimeric, binds to the promoter regions, the consensus sequence response element or regulatory element on responsive genes. Target genes is a term that I had used. Responsive genes, so estrogen responsive genes, you would suspect that the promoter regions on those genes have the estrogen response or regulatory element. So you have some cells, you expose them to estrogen, you look at all the proteins before and after the estrogen. Some of the proteins go up or down. Those are gene products, so they are the genes for all those proteins that change are estrogen responsive genes. Those genes, promoter regions, become candidates to investigate whether they have the estrogen response element. This is the way that we think about it and the source of the terminology. So they may increase or decrease the rate of transcription on those target or candidate target genes. As I said, you look for protein expression that went up or down, and the genes for those gene products are candidate genes, and so on and so forth. So the mechanism of action is to modulate gene expression in the target cells. We've known this all along, and we begin to focus on it now, and we get into greater and greater detail as we proceed with our considerations of all the neuroendocrine axes. So by selectively affecting only at that level of transcription on many potential candidate genes, in some studies they may only look at one gene product, but in reality there are many gene products that can change, and there are some methods. You may have heard of the term DNA microarray. It's a a method of looking at thousands, tens of thousands actually, of messenger RNAs and look at how estrogen would have changed those message levels. So now you're looking at message rather than protein, up and down, but it's the same way. That's called genomics. You can also look at large numbers of proteins with some new methods called proteomics. Anyway, some studies will focus on 1 or 2 genes, but in reality, we expect many genes to be affected by these steroid and thyroid hormones through their receptors. So we would expect message to change, protein levels to change, and if we change proteins, it changes the phenotype of the cell. So I like this one because we've always talked about the response of the target cell. Now we think of phenotype. So the cell looks this way and is doing this thing. That's its phenotype. If that changes, we didn't change its genotype, we changed its phenotype. So it's the utilization of a term we're familiar with, not so much in thinking on the level of gene expression and protein levels and biological capabilities of a cell based upon the functional proteins that are present within it at any one point in time. So now let's consider the structure of these intracellular receptors, these ligand-dependent transcription factors. So we said they're large group transcription factors, so there are some generalities for all of them, just some slight variations in amino acid sequences, but overall we're safe in looking at them all as if they are a single type of transcription factor. So there might be multiple forms of a given receptor that would be expressed in any one particular cell, and we'll see one recent reference looking at 3 different forms of the estrogen receptor in anterior pituitary cells. So it may be from different genes or from alternative splicing, and we'll wait to see when we get to that point whether it's each of those or one of those or both of those cases. So it adds to the complexity of the overall response of the target cell. And it's important to keep this in mind too, because if you're performing a study and something is added and it doesn't produce the clear-cut response that is predicted from the hypothesis and everything you know about how it should function, you have to stop and think that there's many other proteins, there might be other forms of the receptor in there, and we have a different level of modulation depending upon whatever other biologically active proteins are in that cell. So we need to keep that in mind. So they're composed of just a single polypeptide chain, so there's no complexing with other subunits. And in the simplest analysis, there's 3 distinct domains: the amino terminus and carboxy terminus and the things in between. So this is rather routine, as we know, for considering the structure of proteins. And there can be more sophisticated and complex ways of evaluating them, as you will see, especially for the membrane receptors that are much more complex in their overall structure. So this region is involved in activating or stimulating transcription, and we'll see an example of that as we proceed. It might interact with other components of the transcriptional machinery. A large number of proteins are there and contribute to the probability that RNA polymerase will bind to the start site on the gene to initiate transcription. So many proteins involved, as you most likely know. And this can vary tremendously between the different receptors in the subfamily. Then there's a DNA binding domain in between the N-terminus and the carboxy terminus. Excuse me. So we see that there's going to be some type of complex folding of this receptor and conformational change to expose this DNA binding domain. So there's a collection of amino acids that are responsible for binding of the receptor to very specific and rather small sequences of short sequences of DNA, as we've seen in some examples already, and we'll see more. So there's a DNA binding domain in the carboxy terminus,
is typically the ligand binding domain. So ligand binds to one end, and there's conformational change. The DNA binding domain is exposed, as we'll see, and then the amino terminal end is available then to interact with other transcription factors. So here we see it in a structure, yellow, red, and green. Carboxy terminal, where the terminal and the portion, the amino acid sequences closest to that, are committed to binding to the ligand, the hormone, and the middle portion, we'll look at that in a little more detail in a moment. And then the opposing end, of course, is available, sticking out there somewhere, to interact with other transcription factors, including binding to form the dimer, whether it's homodimer or heterodimer. This image is from that article that I pulled up at the beginning of this lecture, so it is available on the link on our website. And it shows a little more detail about the receptor. In this case, it is a typical steroid receptor, but we're going to continue to discuss this more so thinking of it as the progesterone receptor protein. So we see that we add one more component in the mid portion, D, that's the hinge. So if there's a hinge protein, obviously this molecule is going to be changing shape, and obviously in the ligated form it's going to be in one shape, and in the unligated form it's going to be in another shape. So the hinge region obviously is going to contribute to a variety of functions of the other parts. So it's A through F. We can see that D and E, some of D and some of E, are committed to hormone binding. The DNA binding portion is here, and we begin to ask ourselves, what is this figure really illustrating? How do we know any of this? Of course, they produce mutants of this. Substitutions is the most likely way of mutating, or possibly deletions. So if the C region set of amino acids, some or all of them are not there or changed, you can ligate the hormone, but it will no longer bind to DNA. So there's methodological approaches that have been used to determine the function of these. So if you only have A, B, and C, it won't bind the ligand. If you only have D and E, C, D, and E, it'll bind the ligand, but you don't get any effect, even though it might even still bind DNA. So this is the basic approach of determining what these are for. So to get any activation, you can get DNA binding with just the C and some of the D region, but you don't get activation unless you've got the ligand binding portion and some of the N-terminus. And then the C and the D component are important for nuclear localization. So we stated way back when we talked about the cell, we illustrated the nuclear envelope with the nuclear pores and the pore proteins, and there are guard proteins there. And if you don't present certain molecular features that are located obviously in the C and the D region, the DNA binding domain and the hinge domain of these receptors, if it's not exposed and presented, you don't get permission to translocate to the nucleus. It's all diffusion. Diffusion and random collision. There's no purposeful movement of these ligated receptors to the nucleus. If they reside in the cytoplasm originally, if they're always synthesized in the cytoplasm, and if they become nuclear to reside there and wait to be ligated, this is the region that is responsible for it. So there are two cases. Might be in the cytoplasm, can't get into the nucleus until it's ligated and presents the permission, the appropriate part of itself to gain permission to the nucleus, or it's synthesized in the cytoplasm on the free ribosomes and diffuses into the nucleus because the permission is granted based upon this portion of the protein. So the binding and the interaction with the DNA, so obviously steroids are lipids, They enter the cell, simple diffusion. They're in there bouncing around, diffusing around. They bump into the C-terminal end of their cognate receptor and bind to it. It's ligated. Then something can happen. I said the thyroid hormones enter through facilitated diffusion, but still they gain access to the inside of the cell. They're either the receptors are either in the cytoplasm or the nucleus, and that's where they meet the hormones. So I say they reside in the cytoplasm or they reside in the nucleus. So that's the terminology that I will use to indicate where the unligated receptor is located. And obviously, upon ligation, it's going to be in the nucleus, ultimately, in either case. One will translate to the nucleus in the unligated form, and the cytoplasmic receptors will only translocate to the nucleus after ligation by the cognate receptor or ligand. So the hormone binds to a receptor, and the characteristic series of events occur. You get receptor activation, which is the term used to describe the conformational change. So you've got your hormone receptor, it's ligated, the hinge region is going to change, and it's going to reconfigure to expose new parts of the DNA binding domain, maybe the nuclear permission sites, and definitely the N-terminus for interaction with other transcription factors, forming dimers and interacting after binding to the promoter region with other transcription factors. So the major consequences of that activation really is that now the DNA binding domain is exposed. So if it happens to encounter its consensus sequence on a piece of DNA, then it can bump into it. Again, it's diffusion and random collisions. There's no purposeful movement, so there's no cytoskeletal component that says, ligated receptor, pull it here and stick it there. It's all diffusion and random collisions. We have to keep that in mind at all times, even within the nucleus. So the activated receptors now can bind to the hormone response elements. So HRE will be the term for these generic hormone response or regulatory elements. They're short, specific sequences of DNA and are located in the promoters of the hormone responsive genes, as we can conclude on our own by now. And in most of the cases, these ligated receptor complexes will bind in dimers, as we said, and we'll see that in a figure in a moment. Then the transcription from the genes to which the receptor is bound is going to be affected, either increased or decreased. And we'll talk about the transcription factors having different affinities for their response, the consensus sequences of the response elements. So it might be a low affinity and not bind to it. It might be a high affinity and stick to it, with a lot of integrity, and maybe persist there for a long period of time. And then it may also bind with a low affinity and have another effect, as we will see, all affecting transcription. So as you might expect, there's a number of variations on the themes as we've been describing. So knowing about how a specific receptor functions is the important distinction. Not all of them behave exactly the same, but there are many common features. So in the absence of a hormone, some will bind to the hormone response elements, the HREs, loosely with a low affinity, and silence transcription. So this is one possibility of having the target gene silenced. And then when it's ligated, then you get and the receptor becomes activated, then the affinity increases, and it can strongly stimulate transcription. So unligated low affinity binding will decrease or silence transcription. Ligate it, change the affinity with which it's bound, and it now has a dramatic effect on increasing gene expression. Some will bind the DNA not with another of its own kind, but with a different intracellular receptor, of course, forming a heterodimer. In this case, we're going to look at a homodimer. Their illustration shows the glucocorticoid receptor in blue and in green, just to show that there are two of them there that are binding together close to the N-terminal region. And we can see a piece of DNA, and the DNA binding domains of each of them are sticking to the DNA. And somewhere would be the ligand binding domain, the carboxy terminus end of this. And in this illustration, they have not illustrated the ligand. So you can see the actual binding to the DNA. I think by the time you would get the binding of these, the double helix would have been loosened already in this area, but maybe not. This might be how we can see that some of these transcription factors can contribute to loosening the tight coil of the DNA double helix. So the consensus sequence of the hormone response element in this case, called a glucocorticoid response element, or GRE, is GGTACA, and then the N means that there can be any 3 nucleic acids there, and then TGTTCT. So you can see that if you took this and flipped it back, that these are complementary to each other. So they are anti-parallel in sequence and are the opposite. So starting here and going that direction, you basically have the complement starting here and going in that direction, similar to an anagram type of a situation. It's not the exact word I'm looking for that phenomenon, but I haven't been able to remember it all semester. Someday I will. So the N can be any nucleotide. So I'm expecting that you'll be able to recognize this as the sequence for the glucocorticoid response element, anticipating that it would be a fill-in-the-blank type of a question where the sequence would be presented and you would have to recognize it as the glucocorticoid response element, or typing in GRE to get credit for the question. Well, this illustration is also from that review article that I showed you at the beginning, and it is considering different mechanisms of activation of steroid or thyroid hormone receptors, still considering them quite generically. In the upper panel, what we see is that the unligated receptor is complexed to chaperone proteins. These are heat shock proteins, and they are chaperoning this protein, not just letting it float around in the nucleoplasm in this case, so preventing it from being degraded or eliminated any other way. It is available to be ligated. Once ligated, it undergoes a conformational change. In the unligated form, it has a high affinity for the chaperone protein. In the ligated form, there's a conformational change, so it has a low affinity for the chaperone protein and lets go of it. Now it's available and, of course, has a high affinity for another ligated molecule like itself, and it can form a homodimer, and now finally can bind to the hormone response element and affect the transcription of the target gene. There's another case where these Rx receptors can bind to the unligated steroid receptors in a heterodimer, producing a low affinity. When the hormone receptor of this heterodimer is in the unligated form, there's a low affinity binding, and it can silence the target gene. When the hormone receptor portion of this heterodimer becomes ligated, changes to a high affinity binding, and can have dramatic effects on the gene expression increasing transcription. So this is an illustration that makes that point that we talked about earlier. I included the entire figure legend here to help you to look that over and gain a greater appreciation for the diversity of the possibilities of these ligand-dependent transcription factors. So there are some exceptions for the class 2 superfamily. And so in contrast to what we just saw for the thyroid hormone, in the portion of the figure 1, the thyroid hormone, retinoid, and vitamin D and orphan receptors that have been studied all appear to tightly bind to the nucleus at specific sites in the DNA in the absence of the ligand. So there are exceptions to this general rule that we have been studying and were inferred or stated directly in the textbook. So these proteins typically form the heterodimers with retinoid X receptors, the RXR receptors as we saw, rather than the homodimers, and that form these receptor forms recognize sequences that match the estrogen receptor half site, so we can have full consensus sequence matches, the anti-parallel portions that we talked about, or maybe only one half of that sequence is there, and then it's half of a response element. And this is in particular the case for the estrogen receptor, where there are whole ER E sites and half ERE sites. So we're beginning to see a great amount of diversity, and we'll look at those estrogen response elements, whole and half sites, in greater detail in the future. So there are some variation there, and of course the specificity and the affinity of the binding might differ. And the ligand and the DNA binding may also cause dissociation of repressor molecules, allowing association with general transcription factors, other DNA binding proteins, and or coactivators, resulting in transcription of the target genes. So in the unligated form, it allows the repressor to be present and other interactions of the transcription factors to silence the target gene. Upon ligation, the repressor molecules are released, and the altered interaction of the transcription factors in that area to increase the gene transcription rate. There's one other post-translational modification that can occur. All these things that we're considering are still kind of in that post-translational processing. Mechanisms of biological activation and activity are also post-translational processing features if we want to generalize that definition a bit. Definitely phosphorylation, that as is shown here. Again, this is the C-terminus end for hormone binding, the DNA binding portion and hinge portion in the center, and the N-terminal binding portion to the left-hand side here. This is the progesterone receptor. There can be a longer form of it, progesterone receptor B, or a shorter or truncated form of it, progesterone receptor A. At the moment, I don't remember if this is post-translational cleavage or alternative splicing at the level of RNA transcription, but at least we know enough now to ask those questions and to recognize that it would be either RNA processing or post-translational processing to produce a truncated or shortened version where otherwise the amino acid sequence is identical. So amino acid residues 211 and 260 corresponding to the full-length hormone receptor. These can be phosphorylated in the unligated form. When it is ligated, then there can be additional phosphorylation. So this hyperphosphorylation, or this varying degree of phosphorylation, contributes to the degree to which the ligated receptor can activate the target gene. So as shown in that figure, there's the 3 sites on the N-terminus, identified as the region important for transcriptional activation. And the fourth is in the region between the DNA binding and the hormone binding domains, that is the hinge region. And two of the N-terminal sites, the SER211 and SER260, are phosphorylated in the absence of the hormone, and that is increased in response to the hormone treatment, whereas other sites, the SER530 and 367, are detected only after the hormone treatment. So a progression of phosphorylation depending upon the ligated, the degree of ligation of the hormone. So of particular interest is the hinge region site where the SER530 is located. All of the steroid receptors contain either a SER-pro or a threonine pro at essentially the same position, so this is a conserved site. So this may be occurring in other nuclear receptors. In particular, this is just presenting evidence for the progesterone receptor, but because it's a conserved region, then there might be phosphorylation of the hinge region and other of these class of family receptors. This site is also phosphorylated in the androgen receptor, and there's an analogous site that's been reported, at least in the mouse, estrogen receptor. So there's a couple of examples to prove, to support this idea presented there. So the last thing we'll look at in this lecture is how that phosphorylation may be important. Here's the N-terminal portion on one of these ligated receptors, hormone receptors. It's in a homodimer bound to the HRE, and this N-terminal portion that's phosphorylated is available to hold this other transcription factor in place with the transcription complex at the TATA box. In the unphosphorylated form, it cannot hold that other transcription factor in place to impact transcription. So we'll begin with this slide in the subsequent lecture. And, touched the wrong button there. I appreciate your time and attention and hope you're having a nice day.
Thank you.
You're welcome. Bye-bye.